We have Amy and Gira. They are in a bit of a whirlwind because we are the last, I guess, duty before they leave for Johns Hopkins. So they're literally moving lab right now. But they're wrapping up our week, and this is a very topical lecture because right after this, we're going to do a roundtable, but right after that, we'll do a COOT tutorial and a Phoenix tutorial. So Amy and Gira are going to talk about model refinement and validation.
Okay, working? Yep. Yeah, just turn on. Okay, cool. And good, all right. Happy to be here despite all the craziness in our lives. Last time, I guess we'll be doing this probably, Ed, since we'll no longer be local. But we'll be talking today about, so after you've built your model in COOT, what do you do with it? How do you refine it to make the best possible sort of final structure? How do you know that your final model is good? And how can you find areas that still need work? It's too bad that Ali wasn't able to be here today, but I'm also happy to answer any questions, talk about model building stuff too. So I guess afterwards we can jump into all that. Okay, so what's the goal of model refinement, right? So ultimately, we're trying to make the best model we can, so the model that best explains the data we've observed, while at the same time making sense in terms of what we know proteins should look like, right? So we have a lot of prior knowledge about what protein structures should look like. We know how long interatomic bonds should be and what bond angles should look like. So we want to kind of balance these two things, like fitting our experimental data, but also having something that makes good chemical sense. Okay, and so I often like to think of the sort of refinement process almost the same way, in some biochemistry class, you probably saw these diagrams of a protein folding funnel, right? And so you can think sort of at the early stages of refinement, your model is not so good. Maybe it's sort of like a partially unfolded protein, molten globule. And so it's kind of representing a sort of higher energy state, right? And in the process of adjusting the model and refining the model to better fit the data, we hopefully will eventually drive our model all the way down into kind of some kind of local minimum, right? The best possible way we can describe the model. And so in this example of kind of protein folding, on the right side we've got everything from kind of like an unfolded protein molten globule all the way down to the final native folded state, and we can describe this in terms of sort of like the energy of unfolding, right? And so at the top is really high energy protein structures, and at the bottom is the lowest energy state. Okay, and so thinking of this in terms of refinement, again, we've got sort of like different conformational landscape of the protein that's described on this surface, but instead of sort of free energy of folding, in the refinement process, we have some kind of scoring function we're trying to minimize, right? And we can talk a bit more about what that scoring function means, but this is kind of like, I guess, one way you can think about this process in a nutshell, right? We're kind of trying to minimize some sort of function while adjusting the model to try to maximize the agreement between the model, the data, and those other kind of chemical parameters about what we know about protein structure. Okay, and so, in its absolute most simplest form, that refinement target we're trying to optimize could essentially be just some measure of agreement between the model we've built and the data. And so you can imagine you have an experimental map. If you're doing single particle EM, you've got your EM density map. And from a model, we can calculate what we would expect the EM density map to look like. And we could just compare these two and say, okay, so if we change the model and it agrees better with the map, we're on the right track. If it agrees less well, we're on the wrong track, right? This is a very important part of the process. But of course, we also don't want to just move atoms without any regard for chemistry, right? So we have to bring in other terms to this process as well. But I guess before we get to that, so this was just to kind of illustrate, one sort of map, right? And so in a case where you have a map that looks like this, where the position of every atom, every heavy atom is kind of defined by your data, then you really could essentially just have your refinement function be agreement between your model and the map, right? Because your data is going to be good enough to tell you exactly where every atom should be reasonably well. But of course, in practice, we very rarely have maps like this, and this is actually an X-ray map, not an EM map. I have yet to see an EM map that looks quite like this. So in practice, our EM maps might look something more like this, where there's quite a lot of information here, but certainly the map is not telling us the position of each individual atom. And in many cases, it might not be telling us very well the position of many of the side chains, right? And so here's where we have to bring in that additional kind of prior knowledge to guide the refinement process. Okay, and so here's what would happen if we refined our model using only the data, right? So this is just playing around in COOT, saying like, real space refine this with no restraints, just only trust the data. And so when we do this, like that's what our model would look like. So COOT thinks that the best fit of these atoms to that map is this spider web sort of thing, right? So clearly the data alone is not enough to tell us where the atoms should go, and we have to bring in this other prior knowledge. Oh, I think I've got another video maybe with like a whole helix or something. What are we going to do here? Right, okay. Probably playing around in COOT if you've already kind of started working with some of your own data, right? You might have done something like this if you don't have your restraints set properly, right? So the data alone for the vast majority of cases is not going to be enough to refine our model. So we have to bring in other sort of knowledge. And in reality, it's sort of we're kind of striking a balance between our data and other kinds of prior knowledge like chemical information. And it's sort of a continuum depending upon the resolution of the data set you're working with. And so like on the left, if you're at extremely high resolution, like that first map I showed you where we can see a dot of density for each atom, you might be able to rely essentially entirely on your map. But that's obviously a pretty rare case. In the extreme opposite end, right, you have essentially no data and you could be just predicting the structure, right? And so there you're relying essentially entirely on prior knowledge with no data, or perhaps you have a 40 angstrom map, right? And you're essentially just fitting a previous structure into that map and seeing whether it agrees. But most of the time we're in this middle regime, right? Where we have reasonable experimental data, but it's not good enough to tell us everything we need to know. And then we're going to rely much more heavily on this prior chemical knowledge. Okay, so most of the time we're in kind of this regime here. And so our refinement target then becomes a sum of a few different terms. So it's not just agreement between model and data, but also agreement of model with that prior knowledge. And we can start to divide that prior knowledge up into a series of terms, and we can assign a weight to each of those, right? So again, depending upon our resolution and how much we trust our data versus prior knowledge, we can rely more heavily on one or the other. Okay, so what is some of that prior knowledge? I've mentioned some of it along the way. So we know a lot about what proteins look like, right? So we know what the lengths of each bond in an amino acid should be. We know what these various bond angles and torsion angles typically are. So we know how all these atoms should be arranged in space. We know the chirality, right? So we have L amino acids most of the time, not D amino acids. And so we can feed in all of this information to guide the process. Okay. And of course, we know that getting like a little bit larger scale, like moving up from the individual amino acids, we know that proteins are organized into these secondary structure elements. So we know that our protein should be made of helices and beta strands, and we know that there's hydrogen bonds in particular patterns in between the atoms that make up these structures. And so depending upon the refinement software you're using, you can often enforce either particular secondary structure geometries, so you can force helices to remain helices and beta strands to remain beta strands. You can enforce particular hydrogen bonding patterns to make sure that, say, two strands stay next to each other and they don't sort of slide out of register. So that's some of the other prior knowledge we can bring in. We can also bring in sort of restraints from non-crystallographic symmetry. So if you are working on the structure of a higher order oligomer, you've got a homohexamer, say, is your particle of interest. We would kind of expect that all 6 copies of the protein will be similar to some degree, right? So each one is not going to adopt a completely different fold. We know they're going to have similar folds. And we know that even down to the level of side chain rotamers, there's going to be a lot of similarity between multiple copies in your structure. Even if the overall structure is asymmetric, in those 6 chains that are identical, they're going to share a lot of similarities, right? And so in our refinement software, we can restrain those 6 copies to be, well, we can constrain them so that they're absolutely identical. So in some software, you'll see NCS constraints that forces them to be identical. You'll often see restraints instead. So there, instead of being absolute that the 6 chains have to be identical, we can just enforce them to be similar to one another. And that's often more useful because in reality, there may be some subtle differences between the 6 chains, and we want to allow them to diverge slightly. And so NCS can be very powerful where you do have some kind of symmetry in your molecule or your complex. Okay, another thing that actually I think people probably pay less attention to in EM structures, but is being refined under the hood, are B factors or ADPs, atomic displacement parameters. So these describe like the local motion in the protein or sort of errors in fitting of your model to the map. And one thing we know is that, if say you're thinking of a loop on the surface of the protein that's very flexible, right? So the mobility of all the atoms in that loop will be globally similar, and therefore the B factors of all the atoms in that loop will be similar, right? And so we can restrain the B factors of a given atom to be similar to the B factors of the neighboring atoms, right? Because we would expect that one atom that's chemically bonded to the next atom is not going to be wildly more mobile or less mobile than its neighbors, right? So this is also a very important constraint. So B factors can't vary randomly, but they are coupled with the B factors of neighboring atoms. Okay, and then more chemical restraints, right? So we know that atoms shouldn't overlap unless they're bonded with each other, right? So in the refinement process, we can essentially treat atoms as little bumpers, right? And so if they get too close together, we can have sort of repulsive forces that are going to be pushing these atoms back apart and trying to prevent these overlaps. And so that's another sort of restraint we can bring into the refinement process. And so we can build up all these different restraints from prior knowledge. So again, this is kind of like a model of that scoring function that we're using in the refinement process. So our model is being influenced by both the data, but also stereochemistry, secondary structure, clashes, NCS. Some refinement packages will also bring in some kind of force field that either is, like a chemical force field, like charm, for example, that is kind of trying to model the energetics of the system, or sort of more pseudo force, I'm not quite sure, like Rosetta, right, where it's not really a sort of chemical atomistic force field, but it is providing some sort of information about the way the protein should look. And I'm imagining that deep learning is probably going to be plugging into this sort of area as well. And so all of these different parameters, right, are then feeding into the refinement target. And so we can now write this much more complicated refinement target at the bottom right, where we have some term that's representing agreement between the model and the data, plus something about the model versus known stereochemistry of protein, and we can assign a weight specifically to that term. And we can apply a weight specifically to the data term, and the model that is being guided by some sort of chemical knowledge from the force field, and apply a weight to that term, and so on, right? And so our refinement target can become quite complicated as we bring in more kinds of restraints and prior information. And then the goal in the refinement process is to, starting with some initial model that's imperfect, guide it down this sort of energy landscape of conformational spaces, minimize that complex refinement target we've constructed, and try to minimize that scoring function, right? And when we minimize that scoring function, we're sort of assuming that that is going to be the best descriptor of the data, the best model of not just the data, but the best descriptor of the original molecule that we're trying to understand, right? Okay, so we can talk a little bit about how some of this works in Phoenix. Phoenix is one of the most widely used refinement packages. I heard some talk about CCP-EM and some other things just as we were coming in. So there are certainly many programs out there, and you should kind of play with different software packages, I think, and see what works well for you. I really like also this program iSoldi, although I have to say I'm not the most experienced with it, but it's very like interactive, real time. If you have a workstation that can actually handle it, which I think is sometimes a limitation, it's also very powerful. So anyway, so play with different things, but Phoenix is a great place to start. It was very widespread in X-ray refinement and then has been adapted to refinement of EM models. So this Phoenix real space refinement module kind of covers most of what you would want to do for EM refinement. And some of the big decisions you need to make that are kind of related to some of these restraints we were just talking about is kind of up here at the top in this strategy box. Okay, and so we'll talk a little bit about what some of these different parameters mean. Okay, so essentially, we've gone from the restraints, right? The kind of other prior knowledge that we're bringing into the refinement process, and now I guess we're really talking about. Sorry, I sort of messed this up. So the scoring function, right, describes this refinement target, right, that we're trying to minimize. But now the question is kind of like, how do we actually perturb the model to get from this high energy kind of like poor model we start with to the model that best describes the data at the minimum of this energy well, right? And so we can do that, of course, manually in Qt by manipulating the model and trying to adjust it to better fit the map. But then we can use software packages like Phoenix to kind of try to automate some of this, right, and make our jobs a little bit easier. And so these various strategies up here are sort of different ways you can approach this problem in a kind of semi-automated way. And so in the simplest case, you can imagine, you've just got your EM density map, you've got some coordinates from say AlphaFold prediction or some previous structure, and you need to fit them into the map. And so one of the kind of oldest, simplest kinds of refinement is just rigid body refinement, right? And you might even do this in Chimera or something nowadays, but Phoenix can also do this, where you start with a model that is roughly positioned in the map. It's got to be generally kind of close, but essentially just taking that whole set of coordinates, shifting it around in X, Y, Z, and rotating, we try to fit the model into the map and maximize agreement, right? And so you can see on the left, we have these tubes of density that correspond to alpha helices that don't line up with the alpha helices in the model, but by shifting the model slightly, those helices in the model now kind of fit with those tubes of density. Okay, so that's very straightforward. And then I would say the sort of default, kind of most vanilla, sort of flexible fitting type of refinement is this gradient-driven minimization. And so again, kind of thinking of this energy function or this refinement target we're trying to minimize, gradient-driven minimization essentially just allows the model to relax into the closest local minimum. Okay, and so if we start off at this solid red sphere. I guess my pointer doesn't move, that's fine. So if we start off, if our starting model is represented by this solid red circle at the top left, gradient minimization will essentially just allow that red marble to roll down into the lowest local minimum, and then we'll kind of get stuck there, right? So it's good for kind of allowing the model to relax and fall into that closest local minimum, but our global minimum might be quite far away, right? Down here. And so this kind of basic minimization won't be enough to get us over all these peaks and valleys that separate the starting model from the best description of the data. And so we need other sorts of tools that are kind of a bit more aggressive that can help us get across some of these peaks and valleys that separate us. And so with different kinds of refinement protocols, we sometimes refer to something called radius of convergence, which is essentially, how far you can move that starting molecule across this energy landscape to ultimately sample different places in the energy landscape and try to find the global minimum of the function. And so Something like basic minimization has a very small radius of convergence. It's basically just going to, again, let that marble roll down the energy landscape into the closest local minimum. But other kinds of refinement have a much larger radius of convergence. That means they can perturb the model in much more aggressive sorts of ways and help to get it up over some of these kind of energetic barriers or high points in the scoring function towards the target we're actually after, the global minimum. And so some of these other approaches that are available in Phoenix are things like simulated annealing. So this essentially tries to mimic taking your protein, heating it up so it becomes much more dynamic and flexible and can sample all sorts of different conformations, and then allowing it to cool down. And so you can imagine that by putting energy into the system, right, we're essentially lifting that marble up somewhere on the energy landscape before we drop it back down. And then when we drop the marble back down, it can roll and explore, and it might potentially be lifted up over one of those energy barriers and gain access to some additional sites. So you can imagine if initially we had this model, we've done gradient minimization, and we've kind of gotten stuck in this local minimum. Now we do simulated annealing, we heat the system up, and with some probability the model will be able to surpass that barrier and reach the next local minimum, right? And through some cycles of this, we can potentially explore quite a large conformational space of the protein. Okay. There are other ways to kind of try to explore this conformational space. So another option in Phoenix is grid search. And so this is essentially, like if you were, like, okay, we got to find the lost contact lens in this room, right? We could all sort of like, the floor actually is very nicely made into kind of tiles that are like 2 feet by 2 feet. So we can say like, we're going to check this space, and then we're going to check this space, and we're going to check this space. It's grid search, right? And so we can do the same thing with a protein. And so taking the simplest possible side chain, well, not the simplest possible, but the simplest one that has rotameric states, we can think this serine side chain has 3 possible rotamers, right? And so we can try all 3, do refinement on all of them, and see which one gives the best possible outcome. And so you can imagine doing this with all the side chains in your proteins, and with longer side chains, you'd have more rotamers you need to sample, but you can essentially just try to explore all the sort of rotameric states that the protein side chains could sample, and then see which ones work the best, right? So this, so it's kind of similar idea to simulated annealing, that we're sampling a whole lot of different conformational space, but just doing it in a sort of different way. Okay, and so then taking this back to the energy landscape, you can imagine this is essentially like grid point 1 we're trying, grid point 2 we're trying, grid point 3, and after we've kind of created all those different models, we can refine each one and see, all right, so which one drops down into the lowest energy state that we can sample. And so again, this has a larger radius of convergence than simple gradient minimization, and allows us to explore the landscape a little more thoroughly. Okay, so we're moving kind of towards increasingly more aggressive refinement strategies, I would say. And so something that has quite a large radius of convergence is this procedure called morphing. And so effectively what this does is it can take whole loops or elements of secondary structure from your model and actually try to shift them as a rigid body into a new location. And so what is illustrated here is, so the blue model would be your starting model. And you can see that, I keep wanting to point here, but so the blue model in this area here is not particularly well fit in the map. But we can sort of imagine where this yellow model is could be a better position for that loop, right? And so if we can somehow identify this computationally, we can say, all right, so maybe that blue loop shouldn't be here, but we could shift that whole loop over here, and maybe that would be a better fit. And so that process is called morphing. And so we can kind of do this over the whole protein. We can look for parts of the protein that don't fit the map well, but for which there's additional unmodeled density available nearby that we could potentially shift that loop or that secondary structure element into. And so this allows us to move whole chunks of the protein So it's almost like rigid body refinement, but now we're breaking the model into some smaller pieces and moving smaller pieces. Of course, then this breaks the connection between that loop we just moved and the rest of the protein, right? So this is kind of starting to, it allows us to make large movements and changes in the model relatively quickly, but also introduces some problems that we'll then have to deal with down the road. So some of these things can be a bit of a double-edged sword.
The morphing is applied for a region of the model?
Yes, yeah, just for a region, yeah. Did you have a question back there as well? Or?
I was going to say, like, so after you do morphing, do you have to go back to rigid body refinement or like one of the smaller gradients and conversions?
Yeah, so yeah, you can mix and match these various protocols by checking whichever combination of boxes you want to use. When you're running Phoenix from the command line, I think it can be a little more clear, but there's a lot of defaults kind of, when you're using the GUI, there's a lot of kind of default set that you don't immediately see what's going on, although you can access a lot of this from these other menus. But by default, something like morphing or simulated annealing might be done, say, just in the first cycle, but there might be, say, like 5 macro cycles of refinement, often 3 or 5. That's also a parameter you can change, but it might be that in the first cycle you might do an initial minimization of the model, then you might do something like simulated annealing or morphing or one of these more aggressive things, and then it will usually be followed by a few additional cycles of just gradient minimization. So you can also break this up yourself if you want maximal control and say, okay, I'm gonna run the gradient minimization just one cycle, and then I'm gonna do something like simulated annealing one cycle. But by default, usually there will be either 3 or 5 macro cycles of refinement done, and in each of those macro cycles, you'll almost always do gradient minimization and maybe B-factor refinement, and these more aggressive things might be done just in, say, I think usually the second cycle. Okay. But yeah, it's definitely something you can control, and you do want to mix it up. So after, because like we just said with the morphing, in moving a loop like this, you're going to break the connections between that loop and the rest of the protein, and you're going to need some further refinement or manual adjustment to kind of reform those connections and restore good geometry in that area. And the subsequent cycles of gradient minimization tend to do reasonably well for that, but it's definitely something you're going to want to kind of check your model all over between each round of refinement. Okay, so we're doing good. Right, okay. So I guess, so there's lots of things you can choose from here, right? So we talked about global minimization, rigid body, we talked about grid search, we talked about morphing, simulated annealing. I would say the vast majority of the time I would have minimization global checked, because you usually want to allow the model to relax. You've tweaked some side chains. Usually when you interact with the model manually, you will end up straining some part of it, and this allows the model to relax. You pretty much always want to be doing ADP refinement, refining those B factors. And then these last two, I would say, are kind of optional, but leaving them on by default all the time probably wouldn't matter. Occupancy refinement essentially means if you have some group of atoms that are there not 100% of the time, you can refine the fraction of the time that those atoms are there. So this would most often apply to, say, if you have a ligand bound to your protein. It's very common that the ligand might not be there 100% of the time. Maybe it's there 90% of the time. Maybe it's there 50% of the time. So we can set the occupancy to 50% if we think that it's there 50% of the time, and that can sort of help how we model these things. Also, if you have alternate conformations, right? So you have a rotamer that's in state A 50% of the time, state B 50% of the time. Exactly what that ratio, whether it's 50-50, 60-40. So when this box is checked, Phoenix will kind of automatically be optimizing those occupancies. And NQH flips, this is essentially, glutamine side chain, right, has like one oxygen atom, one nitrogen atom. There's a proton on one and not on the other, right? And so You can manually, when you're working in COOT, try to decide which way that side chain should be flipped to optimize hydrogen bonding with nearby groups. So when this box is checked, Phoenix will try to make those decisions for you. Though especially for any residues that are really critical, say in the active site of your protein, you're probably going to want to be making that decision yourself. And I would say that Phoenix tends to, I think there's lots of cases where it's ambiguous which way it should go. And if you care, I would say you might use this in the early stages of refinement, but turn it off later. Because if it's ambiguous, every time you run refinement, Phoenix might flip your side chains back and forth. And once you've decided which way you think that side chain should go, you might not want Phoenix to be kind of scrambling these at random. So anyway, so I guess actually that kind of gets us to a bit the next thing I want to talk about, which is which of these parameters should you use at which stages of refinement? Because refinement is refinement and manual model building are an iterative process. It's not like you will build your model in COOT and then you do refinement once and it's done. You'll often go through multiple cycles of this where you'll adjust the model, maybe you do kind of some coarse adjustments, then you do some initial refinement in Phoenix, then you have a better model you can go back through, you go through residue by residue, check the agreement of each side chain and each part of the protein with your map, make more adjustments, and then take it back into Phoenix and do more refinement. And so the parameters you choose for refinement in Phoenix for each of these cycles might be a little bit different. And so some of the things you might have in mind in kind of trying to decide which of these parameters you want to select early versus late, there's many factors that can feed into this decision. And so I think one of the most important one is how good is your starting model and how much do you trust that. So you can imagine, say you have your starting model is 1.5 angstrom crystal structure, and now you have a 6 angstrom EM map, and you're fitting this crystal structure in, right? I would trust that crystal structure a lot more than my EM map, at least at the level of sort of side chain positions and hydrogen bonding in the protein, right? So you might not want to use some really aggressive refinement parameters in Phoenix like morphing or simulated annealing, because what you'll essentially do is you will scramble the protein, you'll heat it up, you'll let it jiggle all around, and the map won't be of sufficient quality to pull the model back into the best possible configuration, right? Because you'll have sort of essentially lost all of the high resolution information that was stored in those coordinates. So in some circumstances where your starting model in many ways may be better than your data, you might want to just stick with gradient minimization. And you might want to have a lot of restraints that keep the model from changing too much. On the other hand, if you're starting with what could be a relatively low quality model, so maybe it's based upon another protein structure, but it's only 50% identical to yours, or it's an AlphaFold model, which may be pretty good, but may have, some areas that are not modeled so well, or some errors. Then you might want to be a little more aggressive in your initial stages of refinement and use things like morphing and simulated annealing to help sample conformational state and different conformational states and minimize the amount of kind of manual rebuilding and work you'll need to do. Those are some of the factors. I mean, one other thing I also keep in mind is so early on in refinement, I think it can be reasonable to use some of these really aggressive refinement protocols like morphing and simulated annealing. They're good for kind of this global search of conformational space. But as you get into the fine-tuning stages, you tend to want to stick with milder things like just gradient minimization, because once you have the protein in the right sort of general conformation and you're kind of just trying to get things like side chain rotamers right, you don't want to heat your protein up to 5,000 degrees and let it jiggle all over the place, because all the hard work you've done to precisely position all of your side chains will then get lost, right? So the parameters you use in refinement, some balance of the quality of your starting model versus your data, and also, I guess, whether you're early on in the refinement process or kind of in the finishing and polishing stages, okay? And so again, gradient minimization, you're kind of always safe with that, ADP refinement, occupancy refinement, and in the early stages, you can consider some of these more aggressive protocols. Okay, so maybe this is kind of similar to what we just talked through, I guess, right? So I do always start with rigid body refinement. Often you've already fit your model pretty well into the map using something like Chimera. There's not really much risk in using rigid body refinement. It's never going to, drastically move your model out of the map or anything. So, so I'd usually start off with something like rigid body refinement. Depending upon your protein, right, each domain could be a rigid body, or an entire chain could be a rigid body. And yeah, then you can pick amongst these various parameters. And then in late stages, like I said, kind of go easy, just kind of keep it to the basics. Don't do anything too aggressive. Okay, so after setting your refinement strategy, then we talked a lot about all these different restraints we can apply to the problem, stereochemistry, bond lengths, all of these things. A lot of this will be just kind of baked in by default, and Phoenix will kind of try to optimize the various weights, how much to rely on the data versus stereochemistry. But there are some things that you might want to toggle on and off that you have to turn on or off manually yourself. And again, I strongly prefer running Phoenix from the command line myself, just because then you essentially have this command file that has all the parameters, and you can just scan through and decide what you want to change. All of it is accessible through the GUI, though, and the highlights are here kind of on the main page. We talked a little bit about whether if you have multiple copies of the same chain in your protein, you can hit the use NCS box, and Phoenix will automatically try to identify chains that have similar sequences and restrain them to be similar to each other. Secondary structure restraints, again, so that would be, so when Phoenix reads in your PDB file, it thinks that this is a helix, it will enforce it to remain a helix, and it'll enforce beta sheets to remain beta sheets. So all that will happen kind of just automatically, unless if you check these boxes. If you are having problems, you can manually edit, what is defined as a helix. If you have some area that you think should be a helix, but it keeps unraveling, you can specify, okay, that should be a helix in an input file. Reference model restraints is something I think a lot of people don't know about, but can be very useful. So in that case, where you're starting with a 1.5 angstrom crystal structure and you're in a 6 angstrom map, you essentially, I would say most of the time, you want that input model to stay almost the same. And so what reference model restraints is you can either restrain the output structure to be very similar to the input structure, or you can supply a separate PDB file and say, my model that I'm refining should stay similar to this. And so you can input multiple reference models, say to match different domains in your protein. But so this can be really powerful, I'd say, especially at these cases where you have a really good starting model and your data is kind of modest. There are some options to adjust weighting terms and whatnot yourself. And then down here at the bottom are some additional restraints that people commonly use. I mentioned earlier NCS constraints versus restraints. As of last time I checked, Phoenix might only allow you to have NCS constraints, so it forces the chains to be identical to each other. Although I imagine at some point that'll change if it hasn't already. And then two other very popular kinds of restraints people like to use, rotamer restraints. So this would just restrains your side chains to be preferentially in the kind of preferred rotameric states for each side chain, as opposed to, so about any rotatable bond, there's kind of preferred positions, 0 degrees, 120 degrees, 240 degrees. And so this will kind of restrain your side chains to those most preferred parts of the torsion angle space. And the final kind of restraints that are, I'd say, somewhat controversial, and I would say use with caution, are the Ramachandran restraints. And the reason why these are maybe a little controversial is because we often use the Ramachandran plot as a sort of diagnostic of whether a model is good or not, and whether there are problems with a model. And so we'll look at the Ramachandran plot in a minute here, but if during refinement you have already restrained your protein to the allowed regions of the Ramachandran plot, then the Ramachandran plot is no longer really a great sort of validation tool or diagnostic for areas of our structure that need more work. And so for that reason, I would probably suggest not using Ramachandran restraints in the early phases of refinement, because it can be useful to be able to look at your Ramachandran plot, see, oh, here's this loop where I have a couple of violations. You can go back in and fix all those things. And maybe once in the late stages of refinement, when you've fixed up all you can, then you could potentially turn on Ramachandran restraints to kind of just help keep good geometry of the model in the final rounds of refinement, but maybe not use it all the way through. Okay. I think, as usual, I've rambled on probably more than I should have. I was supposed to talk about model validation too. And so maybe just briefly, so I think many people often think of model validation as something that happens at the end, and it's really just like, oh, is my structure good, and should I be able to publish in this journal, and should people think I'm a good structural biologist or not? But it's something that really you should be looking at a bit through the building and refinement process, because a lot of the tools for validation are kind of meant to help you find problem areas in your structure. And so they can help you identify, every time I do refinement, I take the model, I load it into Qt, and I go through residue by residue, and you should do that. But sometimes visually, as you kind of snake through the protein and you're looking for fit and agreement with the map, sometimes you just won't notice areas that are strained or contorted in some way. And things like the Ramachandran plot and other metrics can help you identify areas that you need to work on a bit more. So it should be a sort of active set of tools you use during the model building and refinement process, as well as a sort of final check at the end that helps you to see how your model compares to other structures out there, and say, like, reviewers of your paper, allow them to assess how your model stacks up. Okay, so there's lots of different metrics we can look at. Some are kind of overall quality indicators of your structure, either geometry or agreement with the data. So things like, correlation coefficients between the model and the map, RMS deviations of bonds and angles, these sorts of things. Molprobity scores that tell us a lot about clash and geometry problems. Some are kind of more local and tell us about things like problems with the backbone, problems with the side chains, and so on. And so we talked a little bit about Ramachandran plot already, so that is really telling us about problems with torsion angles in the protein backbone. Things like cis peptide bonds, which we don't normally expect to have very many of in the protein, those can tell us about problems with the backbone. And then there are metrics looking specifically at problems with side chains, so things like side chain rotamer outliers and C-beta deviations often tell us about kind of problems with the side chains. Okay, and then we talked a little bit about how B factors and ADPs should be constrained such that a B factor of one atom should be very similar to a neighboring atom. And so when these restraints break down, we'll often see examples where there are kind of excessively high B factors or discrepancies between bonded pairs of atoms that can often tell us, oh, say the backbone is in the map, but the side chain is sticking out, right? And that would be why there might be some major discrepancy. So these can help us find problem areas in our structures. Okay, so I'm not going to walk through all of these, but these notes will be, I guess, available online afterwards. So some of my favorites, I really like the model map correlation coefficients to kind of understand how well a structure agrees with the map overall. There's several different flavors of these, and they're described in a lot of detail in this review down here. So this will also be in the notes, and I would encourage you to read this. This paper is all about sort of validation of cryo-EM maps and models. So we know that, I said that we know what atomic bond lengths and bond angles should be. So we always look at these parameters. So this is the root mean square deviation of our bond lengths and bond angles versus what's known from known high resolution protein structures. So we want to make sure that our model agrees well with this prior chemical knowledge. And so these are some sort of typical ranges you might expect your RMS bonds and angles to be in your refined protein structure. If they're much outside of this range, it could indicate a problem either in the building itself or in the refinement process. If your number is much lower, it might mean that your restraints are too tight, actually, and so your model is not able to adjust. You're enforcing only geometry, and you're not really letting the data move the model much. That's pretty rare. More often, your RMS values might be high, and that would usually indicate that you aren't applying enough restraints. You're trusting the data too much, and it's starting to kind of pull your model apart. So these are important kind of global parameters to look at. In the later stages of refinement, there's lots of cool tools in Qt that can be helpful. I really like this unmodeled blobs tool, especially as you start getting to higher resolution. Often proteins will pick up small molecule ligands either from the cell when the cell you're isolating the protein from. It can pick up small molecules from your buffer during the purification process or whatever is in the buffer in your final sample. And so sometimes you can discover something cool and unexpected here. And so I always go through this unmodeled blobs finder, and you can specify kind of at what contour level of the map you want to look for things. You might have to adjust this based upon the noise level of your map and how down into the weeds of unknown densities you want to get. But you may find a lipid bound to your protein or a peptide, some unexpected ligand, ATP, whatever. So sometimes there can be cool things there. It can also help you if there's, some, say for example, you have a loop that appears to be disordered, but actually is like reaching out to some other part of the protein and interacts, right? And there's a part of it that's ordered. So this potentially can allow you to identify parts of protein interactions that, as far as you can tell, is like, here's my domain, and you just see the loop go up and disappear, and it's disordered, but it actually reaches across to the other subunit, and there's 10 residue peptide here that is ordered, and you can potentially identify that unmodeled density here, and then eventually figure out where it came from, right? So anyway, that's a very useful kind of tool to validate here at the end. I didn't even remember I had a movie here. I think this actually was a case where, so this is a little bit of a helix here that, so it was a bit of some part of the protein that is ordered, but has kind of like reached across to a flexible loop that the tip of it interacts with the protein elsewhere, and we're able to see that. Sure, many of you know Molprobity. It's really great, kind of global validation tool. I really like running it through their website. So you can get a lot of these stats just through Phoenix as well, or the PDB validation server. But what I really like when you run it through their website is these percentiles you get for some of the key metrics. I find those really helpful because, so the only thing is your PDB file has to have the resolution line, which depending upon where your PDB file is, it might not be there. But if you look in some other PDB file, search for resolution, you can just copy that line into your PDB file and specify the resolution of your structure. And then those percentiles will be telling you how your clash score, for example, compares to all other structures either across all resolutions or in your particular resolution bin. So I think in this case, the coordinates I put in didn't have that resolution line, so it's comparing to all the structures in the PDB. But if your resolution line is there, it'll tell you the percentile, how your structure compares for kind of structures of similar resolution. So I think especially when you're getting started and maybe a lot of these scores don't necessarily mean anything, it's meaningful to know, oh, my structure is 30th percentile. That might tell you that maybe there's some more work you could do to kind of get, not everyone can be 98th percentile, but you hope to be better than average, right? So shooting for at least, 50, 60 would be good. Okay, what else? So the Ramachandran plot is, like I said, super helpful. All of this is just available through the drop-down menus in Qt. And so I'm going to assume that all of you have come across a Ramachandran plot in biochemistry class or something, but essentially just a description of the torsion angle space of the protein and kind of the preferred areas where particular kinds of amino acids like to reside. And so ideally, the vast majority of the residues in our protein will fall within the allowed areas. I think Molprobity has a target of something like greater than 98% of your residues in the preferred regions of the Ramachandran plot and very close to zero in the disallowed regions. So on the left is an example of a nice Ramachandran plot. everything in blue is in the allowed regions. Qt will adjust the Ramachandran plot to the particular residue type that you happen to have highlighted. And so these blue ones here are actually in allowed regions, but they're glycines. So if we were to select a glycine, the plot would change and you would see that that's allowed. But on the right side, you can see all these red residues, right, that are in the disallowed regions of the Ramachandran plot. So if you pull this up and your plot looks like this, you want to look at those residues and see what is wrong in those areas, right? Because this is telling you that there's something about the backbone of your model that's really constrained in those areas. And Qt makes this really easy because you can just click on each of those red dots, and that'll take you to that place in your model, and you can immediately start trying to figure out what's wrong. And if you kind of think through it, you can figure out which rotatable bond in your protein is phi and which one is psi. So if your residue is down here, one of the simplest solutions might be, well, okay, I'm going to change the psi angle to try to get it up here, for example. Or maybe I'm going to try, for this one here, maybe I could either go up here or I could go here. So it can also help you narrow down like what part of the backbone is having a problem. Okay. Sorry, this is what I was just saying about how on the left, all of these look like they're in disallowed regions, but when we focus in on a glycine residue, you can see the Ramachandran plots change to show you what's allowed for glycine. Okay. Cis peptide bonds are relatively uncommon in proteins because when you have a cis peptide bond, you have some clashes between backbone atoms. So if you have a bunch of cis peptide bonds in your proteins, you should check out each and every one of them and make sure that they're justified by the map. Non-proline cis peptides do happen, but they're relatively rare, so you should visually check each one, and if they're not supported by the map, you might try changing the way you model that. Of course, for prolines, this is less of a problem, and so having cis prolines is not uncommon. A lot of these things are flagged nicely for you in Qt. So where there are cis peptide bonds, there will be this kind of polygon displayed near the backbone. And so here's an example of a cis peptide bond, but it's at the site of a proline. So it's highlighted in green because this is not necessarily surprising. However, if we have a cis peptide bond at a non-proline residue, it'll be highlighted in red. Or if it is not necessarily a cis peptide bond, but a non-planar peptide bond, they'll be highlighted in yellow like this. So as you're going through the model, just visually, if you come across some sort of colored polygon like this, it usually means that Qt is trying to draw your attention to some problem with your backbone in that area, so watch out for those. Okay, what else do I have? Because I think I should wrap up, right, Ed? Yeah, I would encourage you to play around with this validation menu in Qt. You can really catch like many of the problems in your model just by kind of marching your way down through some of these. And there are some that are certainly more useful than others, but I would definitely check out, say, like the Ramachandran plot. I like the unmodeled blobs. Geometry analysis and rotamer analysis will both give you sort of the plots that look like this, where each line is kind of a chain, and these bars indicate areas where there are problems. So just very visually, you can quickly assess, where the outliers are in your protein molecule, and you just click on the bar, and Qt will take you to that residue, and you can try to figure out what might be going wrong. So I think I might just wrap up there. Yeah, and I included some additional links at the end, some additional resources you can check out. But I'm happy to take any questions, chat any more about either building, refinement. I'm sure Gira's happy to chat too.